The whole concept is that synthetic biology allows for the programming of different kinds of cells so that they can sense and respond to different signals in the body. So people are now engineering what they call living medicines. It's definitely changing immunotherapy, cancer therapy, and medicine in general. And the basic idea of how it works is you have a bacteria, and then within this bacteria we have what we call a genetic program that consists of these different genes. This particular set of genes is what we call the synchronized lysis circuit. And essentially what this circuit does is it has two genes. One gene has an enzyme that makes small molecules in the bacteria, and these small molecules can actually travel outside of the bacteria and communicate with other bacteria that are nearby. And then when other bacteria sense this molecule, it actually activates this system. And when that happens, then this E gene here will actually then kill these bacteria and allow them to self-destruct. And the point of that self-destruction is that they can then release a therapy into the surrounding environment. So if you zoom out, these bacteria are inside of a tumor. And we have all these little bacteria. These bacteria will selectively colonize tumors because they like to hide from the immune system. And when they live in these tumors, they will actually be making these small molecule drugs right after they burst open and they hit a critical density. And then these small molecules will then diffuse away from the bacteria and start to interact with the cancer cells here. If we zoom in on these cancer cells, Sreyan will tell you how this works.
What we were trying to think about when we started this project was what are some interesting targets that we can block. So one such example is CD47, and it's this don't eat me signal that's expressed on the surface of several types of human solid cancers. This is what tumors use essentially to hide from the immune system and evade being eaten by immune cells. So what we did was we engineered these bacteria that Tal was just describing to release these things known as nanobodies. So the nanobody essentially will block CD47, and so it makes it easier for immune cells to come in and eat the tumor cell and then kickstart an immune response against the tumor.
There's a couple of interesting aspects about this circuit. The bacteria have to hit a critical mass, and that's when they burst open. It releases the drugs better. And then at the same time when they burst open, they actually release bacteria membrane, which will then stimulate the immune system.
Once we verified that these bacteria were leading to promising therapeutic responses, we wanted to characterize whether there were components of the immune system that were being activated in response to these bacteria.
What Sreyan would do is bring the tumors up to our lab. He wanted to analyze or really assess what the T cells inside the tumor tissue looked like. Whether these T cells were specific for tumor cells, whether they were dividing, which would indicate that the therapeutic was helping to activate the T cells, and one cell gets analyzed at a time.
We observed that not only tumors that were injected with these therapeutic bacteria completely eliminated, we also saw significant slowing of tumor growth in untreated metastatic lesions.
What we're doing in the tumor is awesome, but just we're modulating the immune system, right? We're modulating the immune environment within the tumor.
We combined our expertises in synthetic biology and immunology, and that was definitely necessary because we not only needed to engineer these bacteria, but you also need to be able to figure out what kind of immunotherapies you should make and the effect on the entire system as a whole.